All right, so now we're going to talk about the pyruvate dehydrogenase complex, right? So before we had glycolysis coming in, breaking down the glucose into pyruvate, so there's oxidation process that takes place. And now that pyruvate needs to be reconfirmed into acetyl-CoA in order to enter the Krebs cycle, right? So the way it does it is through this specific complex called the pyruvate dehydrogenase complex, right? So if I start off here, pyruvate now enters the mitochondria, and now it's requiring to be further oxidized into acetyl-CoA. So this whole phenomena leads to this production in order to reach, for example, the TCA cycle or the Krebs cycle. So it wants to go to the Krebs. Okay? Now, what is required? So the first thing that is required is the enzyme. So the enzyme that is required is the pyruvate dehydrogenase. So PDH is required. Okay, and so again, you heard the word dehydrogenase. So in this scenario, there is a specific enzyme that you've been very familiar with, is the NAD positive nicotinamide. So an NAD gets converted to NADH plus an extra hydrogen also. Okay. Also, you notice here in this molecule, so you have this oxidation of the pyruvate that takes place, and also you see that there is a CoA group added. So there's an enzyme called coenzyme A, and coenzyme A comes in and binds, so you see the coenzyme A, SH, and binds to this pyruvate here, while releasing carbon dioxide, right? And so you can see here that this is the basic phenomenon of converting pyruvate into acetyl-CoA. And so this enzyme is highly regulated, right? So what is it regulated by? When it's in its unphosphorylated state, so if I were to draw here, the PDH, when it's in its unphosphorylated state, it is very active. So if it's, you see the PDH, and there is no phosphate group around it, now this is actually active. And you can see this process to take place. However, there is another state where this molecule can actually be inactive. So PDH will be inactive. And this is when it becomes phosphorylated, right? So when we look at the regulation of the pyruvate dehydrogenase, is when you have this phosphate group added to it. Okay? So what causes this phosphorylation here that takes place? This is a pyruvate kinase. So there's a kinase that regulates this phosphorylation, requiring an ATP molecule for this phosphorylation to take place. Vice versa, the removal of this phosphate group is acted upon by a phosphatase. So this phosphatase removes this phosphate group and shunts it outside. So you have this entrance of some phosphate group, whether it's from ATP or some circulating phosphate group, and this is what causes this phenomenon to take place, right? And so there are certain positive and negative factors that actually stimulate the kinases. So when you have excessive levels of ADP and pyruvate, this is a negative inhibitor of kinase, right? This is a negative inhibitor of kinase. So whenever this kinase group becomes inhibited, the pyruvate dehydrogenase remains active. So it's not phosphorylated, so it remains active when the kinase is inhibited by either ADP or pyruvate, all right? However, there's other activators of this, right? So we can write that in yellow here. And very common activators of this phenomenon is the actual acetyl-CoA and also NADH. These are activators of the kinase group. So the positive means activate. So if I draw a little line to separate them so you don't mix them up here. So you have this activator. And then again, this contrast, probably you won't be able to see it differently because the green and yellow probably looks the same. But again, you can see the difference, right? So the ADP and the pyruvate are negative inhibitors of the kinase. So this negative inhibition leads to an actual stimulation of this pyruvate dehydrogenase complex. Vice versa, whenever you have this positive regulator here coming in, you can see that this flat line means inhibition, right? So it's really important to understand that. And then what actually causes the actual activity of this phosphatase here, in order to remove the phosphate group, is calcium. So when you have high levels of calcium, this leads to the actual positive activity of the phosphatase, which means what? Activity or the inhibition of the pyruvate dehydrogenase. So you see an actual activation of this pyruvate dehydrogenase complex created from this calcium. So this is just a very simple, basic regulation of the pyruvate dehydrogenase complex.